Hello, welcome back. All right, so protein synthesis today. So how do proteins get made? Where does the information come from? So there's a lot to talk about here, and I'm going to introduce you to another nucleotide, not DNA, but RNA. So let's just start with that first, and then we can delete this. So this looks like a DNA nucleotide. I'm hoping you can start to recognize it. This is there's the phosphate, there's the sugar. Now the one you have been introduced to so far is the deoxyribose sugar. That's where the DNA gets its name from, deoxyribonucleic acid. This one is a ribose sugar. So it's not deoxyribose. It's got deoxyribose is because it's got one oxygen removed from it. This is just the ribose sugar. And there's our base. So we call this then RNA, which means ribonucleic acid. And these nucleotides, they're stored in the nucleolus, which is another section within the nucleus. Right, so in the previous video, we looked at DNA replication, and there's some similarities here, but the end product is completely different. This is not copying DNA, but we're going to still need to access the DNA, and we're going to be doing something different this time. So let's start. Well, let's just quickly a quick overview. When we talk about protein synthesis, there are two stages. Stage 1 is transcription, and then we'll go on to stage 2, which is translation. Okay, so transcription, if there's two things you must stick in your head straight away, it occurs in the nucleus, and the product of transcription is we're going to make a special molecule called messenger RNA or mRNA. All right, so we'll look at how that's done. So when we're trying, in order to make protein, the instructions have to come from somewhere, and that comes from our genes. By definition, a gene is a section of DNA which codes for a particular protein. So if we imagine this is a big long strand of DNA, and this little bit here, there's a section. So that's our gene, and we want to be able to access it because that nucleotide sequence is going to give us the correct sequence of how to put the amino acids together in order to make protein. So the first thing that's going to happen is that needs to unzip, and hopefully you can guess that's going to be the enzyme DNA helicase. Okay, so now we've got the gene has been exposed. Now there's two different strands here. Now, if you remember, it's double-stranded. We've got one here on the left called the sense strand. There it is there. And this one that's been opened up to be what we say transcribed, this is the antisense. Okay, so what we're trying to do here, we're trying to get the information that's on the gene out of the nucleus so that it can be used to put amino acids together. So the sense strand is actually carrying the correct information. It's got our actual sequence that we want, is the gene. So if we want to make a copy of this gene, so there's our strands there, I should put them on. If we want to make a copy of them, well, we can't just do a photocopy. We have to do a complementary, or we have to use the complementary side in order to try and get as close to these ones as possible. So for example, if this was C, then this antisense strand must have been G, which means now when the three nucleotides bind on, we're actually going to get, let's change the color now, we're actually going to get a C here. So the idea is we're trying to get a copy of the sense strands, but to get a copy of the sense strands, we have to use the antisense strands. So these nucleotides are going to bind on. So remember, this RNA nucleotide looks just like a DNA nucleotide, but there's, apart from the it not being deoxyribose, it's a ribose sugar, there's one other really important feature that makes a difference. So DNA, the 4 bases are A, T, C, and G, whereas in RNA, we've got A, we've got C, and we've got G. We do not have T though. We have U instead, which is uracil. So that's just being aware. So whenever you see an A, the complementary, it can't be a T, it doesn't exist, it has to be a U. So if this was an A here, then this would have to be a U. And if this was a T, this would be an A, and so on. So this process of the RNA nucleotides binding onto the complementary bases on the antisense strands, we're starting to produce a single strand. And we call this single strand messenger RNA. So those phosphodiester bonds are going to be formed as well. And now we've got our complete mRNA, and that is transcription complete. Remember, that was the whole point of transcription, was to make mRNA. That's done. So now the instructions are on this mRNA, and this now needs to leave the nucleus. So these little holes here, these represent the nuclear pore. So this is this membrane here, this is the nuclear envelope. So the mRNA is going to leave the nucleus, and it's going to go to where protein synthesis occurs, which is on the, let's do that again, which is on the ribosomes. So that ribosome has 2 subunits. It's got this big one here at the top and a small subunit. And a ribosome is roughly half protein, half another type of RNA called ribosomal RNA. Right. So now we can talk about the second stage, which is translation. So translation, it doesn't occur in the nucleus, it occurs at the ribosome. And rather than mRNA being produced now, this is where the actual protein is being synthesized. So a quick reminder again, protein is if you put lots of amino acids together, that's how you make a protein. So this is all about putting the correct amino acids in the correct order. And if you do that, the intended protein is going to be made. And this goes back to looking at primary structure. The primary structure of a protein is by definition the sequence of amino acids. What sequence have they been put in? And that sequence is going to be dictated by the sequence of nucleotides on this mRNA. So if we look in a bit more detail now, this mRNA on the right here, what's come out from the antisense strand when the complementary nucleotides have joined on, we've got AUG, GCA, AUA, CGU, CAA, CAG, and so on. And they're broken up into triplets, and we call these 3 bases together a codon. So there's codon number 1, codon 2, codon 3, and so on. There's our 4th one, 5th, 6, 7. So we don't look at it now as in individual nucleotide sequence, we're looking at it in codon sequence. So the first codon says AUG, then it says GCA, and each codon represents a specific amino acids. So in order for the correct amino acid to join on, the complementary sequence to AUG, well, what's complementary to A? It's U. What's complementary to U? It's A. What's complementary to G is C. So you can see this one up here has this sequence here, UAC. And whatever we call complementary to the codon, we call this the anticodon. So I'm going to talk to you a bit about this structure up here now. So we've talked about mRNA. This was what got produced during transcription. Then we talked about, which is messenger RNA, by the way. Then we've looked at ribosomal RNA, which is what's found within the ribosome. And now we're going to look at the final type of RNA called tRNA. And that's this structure up here, okay? So it's got this strange shape where it kind of folds in on itself. And these little ladder rungs here, these are representing, this is a big long strand of RNA that has folded up on itself into this unique shape. And that all these parts here, they're not that important for you to know about, but there's two really important regions to know. This bottom part here, which is where the anticodon is found. This is the part which is going to bind on to the mRNA. And at the top here, this is where the amino acid is going to be connected. And it's really consistent. So if whatever the anticodon is here, it will always be the same amino acid that comes with it. So for example, if the anticodon says UAC, the amino acid will always be methionine, always, which is MET. That's a guarantee. So this is important. This codon 1, whenever any gene starts, it's always going to start with AUG, which is what we call the start codon. This is the bit that's going to bind on to the ribosome. It's a bit like if you're trying to zip up a jacket, that bit that you have to connect the two parts together so you can zip up your coat. That little bit at the bottom is really important. Well, that's what this AUG is acting. It's like a priming point for it to join on. So let's quickly draw the tRNA then. This one here is complementary. So here we go. It's UAC have been able to bind on, which means now that methionine is here. Now the next codon says GCA. Well, complementary to G is C, complementary to C is G, and complementary to A is U. So now this one is going to bind on. And this one is called, this amino acid is called arginine, ARG, and so on. So your next one's going to join on. There's our next amino acid join on, which is TYR. It's a bit messy here. Let me just get rid of this here so we can. Our next, I should have made that a bit longer there. Our next tRNA is going to bind on to the next codon, alanine. And as these amino acids are coming into close proximity to each other, they're going to form a bond between amino acids, which hopefully you can remember is the peptide bonds. So a condensation reaction is going to take place, and these amino acids are starting to join together. And if you're sticking amino acids together, you're getting a polypeptide, and a polypeptide is essentially a protein. So once these tRNAs, for example, let's look at this tRNA which is carrying methionine. Once it has connected on and the methionine is joined, and the next one joins up and it drops, and then arginine and methionine can connect together through a peptide bonds. This tRNA here is now able to leave, and it will be able to pick up another methionine for later use in case it's needed later. So these tRNAs, if it's got the anticodon to AUG, which is our UAC, then it's always going to pick up the methionine. It's really consistent with that. And that'll keep going on until we get to the very end codon, which we call the stop codon. And that's where the ribosome will be able to disconnect, and then you've got your full polypeptide. So lots going on here, but let's quickly do a quick summary. Protein synthesis, there's two stages to it. Transcription, which occurs in the nucleus, and it always makes mRNA. Stage 2 is translation. It doesn't happen in the nucleus, it happens at the ribosomes, and the polypeptide is produced, or the protein is produced. There are 3 types of RNA to be aware of. There's mRNA, which is what got made inside the nucleus during the stage of transcription. There's rRNA, which is what's found in the ribosomes. And there's tRNA, which is what carries the amino acids to the mRNA during translation. So, your gene dictates the protein that's being produced. The nucleotide sequence is going to dictate the codon sequence, and the codon sequence is going to dictate the amino acid sequence. And what's the amino acid sequence? It's our primary structure. All right, lots to remember there. Good luck.